Hi, everyone. Welcome you all to the presentation on genetics. Genetics is the latest trend of research. It's the latest version of STEM. Today, I'm very happy to be here in the BIMSSCON 2020 virtual. I'm so excited to see the arrangements of the medical students of Bangladesh, and I'm delighted to see their research activities, their interest for research, and their interest for various areas of research. So today, it's my pleasure to be with you guys and to express my knowledge and my experience on genetics with all of you. So today I'm going to discuss about different aspects of genetics, scopes of genetics, genetic disorders, and the future of genetics. So let me introduce myself. I'm Adnan Mannan. I'm working in the University of Chittagong. I'm an associate professor of Department of Genetic Engineering and Biotechnology. And I studied in Australia. I did Ph.D. in Australia. And before that, I did Masters in Medical Biotechnology from England. And also, I work in Oregon State University, US, as a research scholar of the School of Biomedical Sciences, Natalia Shulzenko Laboratory. And I'm the group leader of Disease Biology and Molecular Epidemiology Research Group, Chittagong. My area of research is genetic disorder, diabetes, molecular epidemiology, and public health. Today, so let me start my presentation. Let me share the slides. Okay. So during this pandemic, during this COVID-19 pandemic, what was the most burning question in Bangladesh? Even what was the most burning question all over the world? Well, in January 2020, what was the burning question? Or in March, when COVID was detected in Bangladesh for the first time, what was the mostly asked question? And that is, where did it come from? What is the origin of SARS-CoV-2? Again, the next question that everybody was asking, how does it look like? Whom does it seem to be similar? Or what's the solution of this pandemic? And the answer is the genome, the gene sequence of this SARS-CoV-2 virus. How? Well, today we'll discuss about that. That's the story today I'm going to talk about. Well, that's the thing. Genetics is the thing that makes all these things beautiful. How? If you consider SARS-CoV-2, the answer of the main problem is go and get the gene sequence of SARS-CoV-2. How can you identify a specific virus? Or how can you identify the evolution of a virus, or the evolution of any animal, or the evolution of any bacteria? It's the gene and genomes. You have to identify the genes in the genome. I'll discuss about gene and genome later. But the answer is, if you can detect the whole genome, you can detect the origin of that virus. How did we locate that? Where did the Bangladeshi SARS-CoV-2 virus come from? When we did genome sequencing of SARS-CoV-2, that time we started knowing who actually came to Bangladesh with SARS-CoV-2 for the first time. And we came to know that it's European originated SARS-CoV-2 genome. This is not the only thing. It is quite exciting. CRISPR-Cas9, the latest genetic technology, another beautiful thing. You can now edit your genome. You can now edit your gene. Whatever you want or however you want, the gene can be like that. That's amazing. That's beautiful. That's the latest application of genetic engineering. Not only that, if you, well, in the clinics or hospitals, most of the times there's a complaint that the baby is missing, or the baby has been exchanged, intentionally or unintentionally. So how can you actually detect the baby, or how can you prove your maternity? That's the DNA test, because DNA has a spatial conserved sequence which is 100% similar in both mother and the baby. And again, another exciting area of research, another exciting discovery is vaccine. A lot of vaccines are now designed targeting the DNA, targeting the gene, targeting the genetic changes. And that is how the world is stepping ahead to get a cure for different diseases, for a lot of deadly diseases. So now the question, what is gene? Where is it coming from? What is the specific area of this term? So before going to that, I'd like to pay a little bit tribute, and I'd like to say salute to these three legends. You know, Francis Crick, we all know these three people who actually discovered DNA. Um, Watson, Crick, and the lady who has been ignored for like ages, and who has been a victim of sexism in the research area, Rosalind Franklin. A big salute to these three discoverers, or these three scientists who actually were showing us who showed us the light, and who is still showing us new avenues of research. And another most exciting thing is the Nobel Prize 2020. In the day before yesterday, we saw in chemistry two ladies, Emmanuel and Jennifer Doudna, got Nobel Prize in chemistry in 2020. You know what it is about? It is about gene editing technology. How that's like, it's quite fascinating. You can edit your gene. You can edit your genome, and Jennifer Doudna and Emmanuel, they actually made it possible. And I had the privilege to visit the lab of Jennifer Doudna back in 2018 when I was visiting University of California, Berkeley. So I believe, I dream that one day people will come to Bangladesh. Bangladesh will have this kind of lab. Bangladesh will have this kind of technology. And that is why we are working about. That is what we are aiming, we're looking for. So where does it start from? It starts from a cell. Well, if you go to Bio 101 or Biochemistry 101, you can find all the answers. Or if you go back to your O levels biology or HSC or a secondary biology book, you'll find the answer. Our body is made up of millions or billions of cells, and in the cell there is a specific region that is called nucleus, and that nucleus actually contains DNA and RNA. DNA it is arranged in chromosomes, and DNA basically DNA contains a lot of hereditary information. DNA contains genome, and also RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. DNA is double helix, and RNA is single helix. So other than nucleus, there is another part of the body who contains a DNA. Mitochondria has a very special DNA, which is called mitochondrial DNA. So that's another DNA, but it has very limited number of functions. So if you talk about gene, genome, and DNA, the first thing that comes to any genetic engineer's mind is genetic code. What is genetic code? Or what is this code? Is that sort of coding or something like that? Yes, it is a code. When someone from CID or FBI or someone like from any agency go to investigate anything, they have some code language. With that code language, they understand what they are actually meaning or what they are actually looking for. That's how they communicate. But other than them, like other people who are not relevant to them can't understand the message. Similarly, our body has a very spatial code, which actually remains in the genes of our nucleus, and a series of messages are actually remaining in the chromosomes. These codes are actually regulating all the functions of our body. Every single codon actually codes for an amino acid, and one amino acid comes from usually one gene codes for one protein, and protein is actually consisting of a lot of amino acids, and three nucleotides of a gene actually codes for one amino acid. So this code actually corresponds to the structure of DNA. This code determines whether you will be black or white, whether you will be angry or you will be very calm and quiet. This code determines whether your hair will be black or golden or white. This code will determine whether you'll be colorblind or you'll be healthy. So this code actually determines a lot of features of the body. So this is the chemical structure of DNA, where we have base pairs called adenine, nucleotides where we have adenine, guanine, cytosine, and thymine. We have a ribose sugar, and we have a phosphate group. So what is gene? What is gene? In DNA, the smallest portion of the DNA is gene. Every single gene contains a specific feature. Every single gene contains is responsible for synthesis of a protein. But sometimes some of the genes don't do anything. They don't have any function. They just remain in the body as a junk part. But other than that, gene is a locatable region in the genomic sequences, which actually codes, which actually controls any work, which actually regulates a lot of work of the body, and it determines a lot of cellular properties. It determines a lot of structural properties. Gene determines what would be the shape, what would be the function, how would it look like. So a lot of features, mechanical features and phenotypic features of our body are controlled by gene. Gene is basically a combination of a lot of nucleotides. So when we consider all the genes together, then that is called a genome. Genome is sum total of all the genes that remains in a chromosome. Genome is identical in all the cells mostly, but not all genes are expressed in all cells. So you might have the same gene in different organs, like in liver, in kidney, in lung. You have the same genome, but the thing is, not all the genes will be expressed in all these organs. Maybe some of the genes will be expressed in lung, some of the genes will be expressed in kidney, some of the genes will express in liver. So gene expression of gene or function of gene depends on the part of the body or the organism of the body. So gene can code for enzymes or different functional proteins, structural proteins, or gene can regulate a lot of activity. So what is actually genetic engineering? It is called a very trendy area of science, technology, engineering, and mathematics. So what is actually genetic engineering? Genetic engineering is basically manipulating genes. It's just, if my term is a layman term, I would say it's manipulation of a gene. If you want to insert a gene in a genome, that is an engineering. So like if you want to insert a new gene in a genome which is missing in that genome, like which is missing in that baby's body, that is genetic engineering. Or if you want to switch off a gene is functioning abnormal that time. If you want to switch off that gene, if you want to insert some materials in your genome, that is genetic engineering. Sometimes we do this gene insertion or genetic modification using bacteria. So bacterial gene is spliced enzymatically, and a specific technology is used that is called recombinant DNA technology, where we insert plasmid into DNA, plasmid inside a body, and that plasmid actually carries a lot of genetic information, which later recombine with the host DNA. So what is gene therapy? When you have a genetic abnormality or when you have defective gene, for the treatment of that gene or for the modification of that gene, if you take therapy, a specific therapy where you can modify a gene or you can insert a gene or you can compensate a gene, you can add a missing gene, you can stop a gene's activity, that sort of therapy is called genetic therapy. And genetic therapy is desperately needed nowadays for a lot of genetic disorders and a lot of inherited disorders. in our country. So now the next question that is coming, a lot of people are now visiting doctors and the clinics and the hospitals with a very strange disorder that is called genetic disorder, that is called genetic disease. What is genetic disease? Where is it coming from? And what is the reason of genetic disease? And what are the types of genetic disease? So first thing, what is a genetic disease? Genetic disease is when your gene does not express protein properly or does not work as it was expected. That time it is called genetic abnormality. Or when your gene is silenced, gene is not working, that is a genetic abnormality of genetic disease. Or when your gene is faulty, a specific part of a gene, say like one nucleotide is missing, or one during your birth, one additional nucleotide which should not be a part of that gene has been inserted by some way, that is called genetic disease. So traditionally, there are three different types of genetic diseases. One is genetically determined, another one is environmentally determined, and another one is occurring as a combination of both the reasons. So today, nowadays, we see 20% of pediatric patients have genetic abnormalities, like they're born with these abnormalities. 50% of spontaneous like abnormalities are happening due to chromosomal aberration, and a lot of genetic diseases are happening where mutations or genetic changes are not lethal. So you carry the genetic change, you carry the abnormality, but you don't die. You suffer for throughout your life. So before going to different genetic diseases, let me tell you some of the terms that we commonly use in genetics. Number one, hereditary. When you get something from your parents, then we call it hereditary. Familial, when transmitted in gametes to a generation. So something is transmitted from generation to generation. Grandmother, then your father, then yourself. That is actually familial. Say like hypercholesterolemia, a lot of people have hypercholesterolemia just because of their family history. Congenital, it's present during your birth when you have some genetic abnormality from the very beginning of your childhood, or from the beginning of your life. That is actually congenital. So not all the genetic diseases are congenital. Some of them are like some of the genetic diseases happen at the end stage or middle stage of your life. So genetic disease doesn't mean that it is congenital or it is familial in all the cases. It can happen because of the influence of a lot of factors. So there are three types of genetic diseases. One is multifactorial inheritance. A lot of factors actually work over there. It's called polygenic. Another one is monogenic disorder. And the third one is chromosomal aberrations. So let's see what which one actually results in what type of disorder. Disorders with multifactorial inheritance. If we talk about this, multiple genes are involved in this case, and also a lot of environmental factors are involved in this case. Sometimes hypertension, gout, and diabetes mellitus, they all can happen together because of a lot of genetic factors or combination of a lot of genes, of like more than one gene's abnormal activity. Sometimes congenital heart disease happen. Sometimes of cancer happens because of multifactorial inheritance, like ovarian cancer, breast cancer, colon cancer. So multifactorial inheritance can lead to a lot of deadly diseases. Gout is another type of disease, which is which is a genetic disease, and it happens genetically impaired metabolism of uric acid. In a lot of cases, uh, uh, usually this sort of metabolism. Incomplete metabolism happens or unusual metabolism happens, where tissue accumulation of excessive amount of urea acid crystals happen, and also large crystalline aggregates in and our joint. This accumulation happens in the joints, so joint deformity is observed in this case. So gout is a very common genetic disorder. Another type of genetic disorder is monogenic, is like mutation in only one gene. It's called Mendelian inheritance. There are 5,000 diseases which happen only because of one abnormality in only one gene. There are three different types: autosomal dominant, autosomal recessive, and X-linked disorders. So autosomal dominant and autosomal recessive means actually um, when when only one change in autism is okay, that's autosomal dominant. But in both the autisms, if you need or both the chromosomes, if you have to have this genetic change to result in genetic disorder, that time it's called autosomal recessive. There are lots of autosomal dominant disorders. Both males and females are affected, and usually heterozygotes, that is inherited from in one parent, mom or dad, and it is transmitted from one generation to the other generation. And 50%, 50% of our children are affected as a result of this disorder. Familial hypercholesterolemia. Familial hypercholesterolemia is very common. It's very frequent. It's a Mendelian disorder. Mutation of gene encoding LDL receptor. It can result in familial hypercholesterolemia. It's sometimes you see that you don't eat a lot, you don't eat junk food, you don't eat you don't eat a lot of food which has cholesterol, but still you have cholesterolemia. Why is it happening? It's your gene, it's your genetics, it's your inheritance. Because of the familial history, you carry the gene, you carry the disorder, and as a result, you're a victim of this disorder. So there are heterozygotes and homozygotes, both of the types of hypercholesterolemia are there. There is Marfan syndrome. French pediatrician Marfan in 1896 actually discovered this disease. Abnormal protein fibrillin, it is secreted by fibroblast because of a genetic change in fibroblast, and it's an extracellular matrix, and there are different clinical manifestations. There is another genetic disease that is called Ehlers-Danlos syndrome. It is similar to Marfan syndrome. Genetic defect of collagen fibrils happen in this case. Hyperextensibility of the skin, hypermobility of joints, these sort of things are happening in this case. And large vessel, colon and cornea, they're ruptured in this case. There is, and the second type of genetic disease or genetic disorder is autosomal recessive trait or autosomal recessive disease, where a majority of Mendelian disorders are seen. Homozygotes are affected, and in this case, we see onset of symptom often happens in childhood, and it's frequently enzymatic defect. Because of this trait, mostly enzymes are not secreted or faulty enzymes are secreted, and for detecting this disease, it is better to check parents' genetic makeup and also kids' genetic makeup. If the mutant gene occurs within low frequency, high probability and consanguineous marriages. If you get married with your relative or cousins, there is a chance of the baby having this disorder. Cystic fibrosis is another type of disease. It's not very uncommon. It is happening. It is like widely observed nowadays. One in every 2,000 live births are coming in the world with this disease. It is a defect in the transport of chloride ion across epithelia. Increased absorption of sodium and water to the blood happens as a result of this disorder. It is this defect is observed widespread in exocrine gland, and our airways are blocked. Pancreatic ducts and biliary ducts are affected as a result of these genetic disorder. Pancreatic abnormalities happen, pulmonary lesions happen, and gastrointestinal problems also observed as a result of these genetic disorder. Another genetic disorder which is also involved with metabolism is phenylketonuria. Absence of enzyme phenylalanine hydroxylase in your body can lead to this disorder. It increases phenyl since birth. Rising level of phenyl is observed. It impairs the brain development. It's your IQ level. You have mental retardation if you have this problem. Um, uh, Early screening test during pregnancy is good. Your diet should be controlled, and decreased pigmentation of hair and skin is observed in this case. And mothers with phenylketonuria have a high chance of giving birth of a baby having this genetic disorder. Another very common genetic disorder nowadays is galactosemia. It's defect of galactose metabolism in our body. Galactose plays an important role. Like milk product or any other carbohydrate, we have a strong association with galactose. Liver, brain, and eyes are affected as a result of this. Cataracts happen due to galactosemia. Um, Usually accumulation of galactose happens in this case in the blood, and since birth people can have this disorder, and your brain can have loss of neurons, loss of gliosis, and also later you can have mental retardation and you can have cataracts. Another similar disease is glycogen storage disease, which we call glycogenosis. It's deficiency of any of the enzymes involved in the degradation or synthesis involved with glycogen, um, which lead to glucose. And also it depends on the type of the defect. There are different types of glycogen storage disease, POM disease, McArdle syndrome, and different other types of diseases are happening there. Another rare disease is Niemann-Pick disease. It's a defect of sphingomyelinase. It's accumulation of cholesterol and sphingomyelin in spleen, liver, lungs, and massive visceromegaly happens as a result of that. And death within first 4 to 5 years, people die after being investigated with this disorder. There are some other diseases who are called X-linked diseases, which is actually involved with our X region of chromosome. X, Y, we have two different regions, and it is transmitted by heterozygous mother to their sons. Daughters are mostly carriers. Sons actually are affected. They are diseased. 50% sons are diseased and 50% are healthy. A very big example of this disease is hemophilia. Hemophilia A is a defect of factor 7, factor 8, and hemophilia B is a defect of factor 9. And hemophilia, in case of hemophilia, female is a carrier, but they are not affected mostly in 99% cases. The main, the main victim of hemophilia is the male. The boy gets it from his mother. So hemophilia is it's an X-linked disease. There are chromosomal aberration. There are abnormality in chromosomal position abnormality happens, and alteration in the number of structure of chromosome happens. We know we have autosomes and sex chromosomes. So if there is the position is mismatched, or there is a mispositioning in the chromosome during birth, or any of the thing is missing in the chromosome, chromosomal aberration can happen. So there are different terms that is used in case of chromosomal aberration and these sort of genetic diseases. One is called euploidy, polyploidy, and aneuploidy. I'm not going to discuss all this in detail because it would be too much for today. But in future, you might come to know about these terms when you come to know a lot of genetic diseases. So there are a lot of structural abnormalities that happen due to this chromosomal abnormality, where deletion or translocation of any position of chromosome can happen. Loss of chromosomal material can happen. Abnormalities of sex chromosome can happen in this case, and also from parents parents you can get it, and also a lot of siblings have this sort of similar chromosomal abnormalities. Autosomal disorder. So we already said that there is a specific part in our cell or in our chromosome that is called autosome. These autosome has a lot of disorders because of excessive number of presence of genes or any specific nucleotide or absence of nucleotide. The most common one is trisomy 21. Trisomy 21 is observed in like one baby in every 700 baby during birth. Parents have normal karyotype, but the baby has abnormal karyotype, abnormal genetic, you know, genetic structure. And it's in the number 21 position. We have three set of chromosomes instead of two. So you can have mental retardation, you can have congenital heart defect, you can have hypermobility, and also increased risk of acute leukemia because of this disorder. There are other disorders that those are involved with sex chromosome disorders. Usually in female, it is observed in female, it happens because of X chromosome, and in male, it happens because of Y chromosome. Turner syndrome is one of the sex chromosome disorders. 1 in 3,000 female has this Turner syndrome, where primary hypogonadism in female is observed. Another very interesting and also unfortunate genetic disease is color blindness. It is sex-linked. Males are the victims mostly. It is present during birth, and it is inability to distinguish certain colors, mostly red and green. So these people can never identify the colors, these two colors throughout their life. Another very common genetic disorder is cystic fibrosis. It's a recessive genetic disorder, usually present at birth. It lacks an enzyme, so mucus builds up in the body, and you can see the fingers, abnormal shaped fingers of this baby. This is a common sign of this disorder. Huntington disease is another common genetic disorder, where in the middle age, you will start getting, or you start being observed having the symptoms. It doesn't have any cure, and these scientists actually discovered this disease. Thalassemia, a very common disorder in Bangladesh, especially in Chittagong, Cox's Bazar, and Chittagong Hill Tract. 40% people are actually thalassemic at the moment, according to our very recent study. So it is a recessive trait. It is present at the birth. It's basically abnormal red blood cell, not having enough red blood cell in the body, and also the red blood cells there that you are having, it might have an abnormal shape. So you need to do blood transfusion regularly as a treatment of this. So now the next question is, how can we actually identify the genetic diseases? So we talked about what is gene, we discussed about genome, we discussed about the structure of gene, we discussed about the types of genetic diseases. Now the next question is, okay, we know the genetic diseases, but how can we actually identify these genetic diseases? How can we diagnose or investigate? There are a lot of molecular diagnoses by which you can see the pattern of a gene, or you can see the nucleotides, or you can see the changes in the gene, or you can see the chromosomal abnormality. One is PCR, polymerase chain reaction, where you can see some bands. If the band is present, that means that genetic specific sequence is present. If the band isn't there, that specific sequence isn't there. That's how we detect a specific sequence, whether that sequence is a healthy sequence, person sequence, or that might be an abnormal sequence. But by this method, we can detect a specific sequence. We have FACS, that is a cell sorter, where we can observe live the movement of the cells or the shapes of the cell live in the microscope, and also a very specific machine. There's Western blot, where we can observe the protein products. If you have abnormal protein, or if you have lack of any protein, or if any protein is synthesized or missing in your body, you can check it by Western blot. You can do gene sequencing or genome sequencing to check whether a gene is missing or whether a gene is defective, faulty. You can do microarray to see the expression level of gene. How much expression is happening from gene? Is that enough, or is that not enough? So amniocentesis or cytogenetic analysis, which is called karyotyping, biochemical activity for various enzymes, we do for this type of genetic tests. We do chromosomal analysis, and this sort of figures we see in this sort of tests. We do karyotypes, where observing the shape and the length of the chromosome, we can check the chromosomal abnormality. Unfortunately, Bangladesh does not have any information for these genetic diseases. Not only these, we found for more than 500 genetic diseases, we don't have any information of Bangladeshi patients. We don't know what sort of mutations are they having. We don't know what sort of genetic changes are common in Bangladesh. We don't know which treatment pattern is okay for this sort of genetic change. We don't know which genetic change can lead to what sort of symptom. For hemophilia, we don't have any single genetic information of any of the Bangladeshi hemophilia patients, which is very unfortunate, and which affects their treatment pattern. Because if you know the genetics, it can help to plan a treatment pattern following the Western or other countries' treatment pattern. Genetic variation, we don't know what's the actual genetic variation among the Bangladeshi cancer patients. We don't know most of the antibiotic resistant gene. Most of the genes, those are responsible for antibiotic resistance in our country. Antibiotic resistance in various bacteria, in human. Which gene is very common? Which gene is most prevalent? Some of the studies of some very reputed Bangladeshi scientists are there, but it's not enough. We need a lot of studies. We need to know a lot of genes. We need to know all the genes or most of the genes who can lead to antibiotic resistance in bacteria, in human body. We don't have any genetic information of the hypertensive patients. We don't have any genetic information that is responsible for obesity, because in the Western countries, a lot of papers are now saying that gene is associated with obesity. So what actually genetics or genetic engineering did so far? Insulin is a discovery of genetic engineering, recombinant DNA technology. Using a bacteria, using a gene, we discovered how to produce insulin outside the body. Cloning, human cloning, animal cloning, plant cloning, it's everything possible because of genetic manipulation. Stem cell therapy, now you're thinking about new treatment methods. Now you're thinking about new therapeutics. Using stem cell, you can regulate the birth of a baby. Now you can using stem cell before the birth of a baby, you can correct the genetic abnormalities. It's a stem cell, and it's genetic engineering. Gene sequencing is the most important part of genetics. Until or before you know the gene sequence, you don't know anything. After you know the gene sequence, you come to know the gene. After you come to know the deletion, substitution, addition, and different genetic changes. And finally, I'd like to say we have miles to go before I sleep, because we have a lot of unexplored area in our country. We have a lot of genetic diseases. We don't know how to cure them. We don't have vaccine. We don't have medication. We don't have any treatment for them. So just like this baby, I'd like to say, give me a world I can grow up in. This baby wants to grow up in, but how would he grow up in if he doesn't have medicine, if he doesn't have vaccine, if he doesn't have therapy? So genetic engineering is the solution for a lot of medical abnormalities. So this is the right time to focus on these science. This is the right time to increase the number of research, and this is the right time to think about a healthier tomorrow, to think about a better tomorrow. So let's make tomorrow better. Thank you very much.